All right, so now we're going to take a look at a little something different. 6.4 is based on viroids, virusoids, and prions, and these are a little bit different. We're not talking about eukaryotic, we're not talking about prokaryotic, we're talking about something completely different. So these are causative agents, so they cause issues, they cause disease, and they're previously uninvestigated diseases, and in looking these uninvestigated diseases, they couldn't figure out what was going on with them, and then they found these non-living disease agents, but they're different from viruses. So they're not eukaryotic, they're not prokaryotic, they're not viruses, which are acellular, but they're classified as non-living disease agents. So these include particles consisting only of RNA or only of protein. That, even though they only consist of RNA or only consist of protein, are able to self-propagate at the expense of the host, so they have to be within a host, which is similar to viruses. That's why it's in the virus chapter here, allowing them to cause disease conditions. So these are three that we're going to take a look at. So first are viroids. So in 1971, Theodore Diner, he was a pathologist working in agriculture, so Agricultural Research Service, discovered this acellular particle that was not a virus, but very similar to a virus, that he called a viroid, so virus-like. A viroid consists only of a short strand of circular RNA, and this just short strand circular RNA is actually capable of self-replication. So it does not have a protein coat, it doesn't have an envelope, it is literally just a circle of RNA. The first viroid discovered was found to cause potato tuber spindle disease, which causes slower sprouting and various deformities in potato plants, and we'll see an image of that in just a minute. Like viruses, though, the potato spindle tuber viroids, PSTVs, take control of the host machinery, and then they replicate their RNA genome using the host machinery. So even though they don't bring anything with them other than this circular RNA, once they get in there, they're able to take control, hijack the host's machinery, and then make copies of itself. However, unlike viruses, they don't have a protein coat or anything around them to protect their genetic information. So here is that image that I mentioned. You can see that the potatoes look deformed, very different. They have these lumps, all of them. The potato spindle tuber viroid. So viroids can result in devastating losses of commercially important agricultural food crops grown in fields and orchards. As you can tell, if they've got these viroids in them, then they're deformed and they're not very useful in the sense that they have these pieces in them, and then we don't get a good crop of potato. They're deformed, they're not healthy, they split apart. You can see the splits in here, so it makes them not a good crop of potatoes, for example. Since the discovery of PSTV, we've found other viroids that cause diseases in plants. So there's the tomato planta macho viroid, or TPMVD, which infects tomato plants. Causes a loss in chlorophyll. It causes disfigured and brittle leaves. Results in very small tomatoes, and so again we lose productivity in this field crop. Also, avocado sunblotch viroid, ASBVD, so lower yields and poor quality avocados. The ASBVD is the smallest viroid that has been discovered so far that infects plants. There's also a peach latent mosaic viroid, the PLMVD. Causes necrosis or death of flower buds and branches, and then it wounds the ripened fruit. And then once it's wounded, then that leads to fungal growth or bacterial growth in the fruit, and then can devastate that entire crop. Also, this PLMVD has been seen and can cause changes in other things like plums and nectarines, apricots, and cherries. So then we can see that this can decimate a field of any of these items. So viroids, in general, can be dispersed mechanically. So this is how we see the viroid getting spread from place to place. So it doesn't have any sort of capsule or any way to kind of burst itself from a cell and spread to other organisms or spread to other plums or peaches or avocados, for example. So what we see is during this crop maintenance or harvesting, or cutting of these things. It can get on the knives, it can get on the machinery that's going through and collecting these things. And once a piece of machinery, for example, that's digging up something, digs up one of the potatoes, for example, that has the PSTV in it, then that would have the loop of RNA on it, the single small circular RNA on it. And then once it does that, then the next time it cuts into the ground to dig up more potatoes, it's going to start transferring that from potato to potato. And then when, for example, in potatoes, once that is transferred to that, the next harvest of potatoes generally comes from a cutting up of the previous harvest of potatoes and then replanting a piece of the potato. And so then, since that potato has gotten that RNA in it, then that would be within the RNA, and then once it's put in the ground, then the potatoes that come from that will all have this viroid in them. So dispersed mechanically. So rather than being spread in other ways, in vitro or in vivo, it's spread mechanically. During crop maintenance or harvesting, during vegetative reproduction, and possibly via seeds and insects, resulting in a severe drop in food availability and devastating economic consequences. So since we have all of this machinery that's going through, then oftentimes we have the same pieces of machinery touching everything as it goes along, and then it can infect the entire crop. So next, we're going to take a look at virusoids. So we have viroids, now we have virusoids. So the second type of pathogenic RNA that can infect lots of crops, or commercially important crops, are virusoids. So these are subviral particles, best described as non-self-replicating single-stranded RNAs. Okay, so our viroids are self-replicating, can make copies of themselves. They get into the cells of the plant, and then they hijack the cell, and then use that to make copies of itself. Virusoids are small pieces of RNA, but they're single-stranded RNAs, and they're non-self-replicating, so they're not able to replicate themselves. So RNA replication of virusoids is similar to that of viroids, but they do require that the cell also be infected with a helper virus. So our viroids can just be all by themselves, get a piece of their circular RNA inside of a plant, and then it can just go along self-replicating. Our virusoids require that the cell also be infected with a specific virus. So we only have 5 described types of virusoids at this time, along with their associated helper viruses. So the helper viruses are all from this same family, the sobimoviruses. So an example of a helper virus is the subterranean clover mottle virus, and it has an associated virusoid packaged inside of its viral capsid. So that's how these guys get around. The virusoids are actually separate from the virus, but they can be within the viral capsid. Once they're within the viral capsid, they have their helper virus right there, and then they have themselves, the virusoids, within the virus capsid. So once the helper virus enters the host cell, then the virusoids are released and then can be found free in the plant cell's cytoplasm. There they possess ribozyme activity, and then the helper virus undergoes its typical viral replication, so independent of the activity of the virusoid. So that's the key piece there, is that it's not a piece of the virus itself, because it's independent of it. It'll be doing its own activity even though the virus is going through what it normally does, its own viral replication. Virusoid genomes are small, only about 220 to 388 nucleotides long, so very small. And the virusoid genome does not code for any proteins. All it does is replicate itself. So it just goes in and replicates itself. And then once it replicates itself, you can imagine we've got this virusoid floating around in the cytoplasm of a cell. And within that same cytoplasm, we have our virus undergoing viral replication. It's going to be making its capsid proteins, capsomeres. And then as it's piecing that together into its capsid, it will accidentally grab a piece of this very small RNA, these virusoids. And then that's how, once the virus then leaves that cell, bursts out of that cell and goes to infect another cell, that's how it carries this virusoid with it. So virusoids belong to a larger group of infectious agents called satellite RNAs. So they're similar to pathogenic RNAs that are found in animals. So unlike the plant virusoids, though, satellite RNAs may encode for proteins. So these virusoids that are specific to plants do not code for proteins. All they do is make themselves over again. But satellite RNAs can encode for proteins. Like the plant virusoids, though, the satellite RNAs must co-infect with a helper virus to replicate. So our satellite RNAs are our larger group. Some encode for proteins, some do not. Those that do not can be virusoids. But all of those in the satellite RNA group must have a helper virus in order to replicate. So one example of a satellite RNA is one that infects humans. And it has been described as a virusoid because of the way it works with its helper virus, but it's the hepatitis delta virus, which can be either called the hepatitis delta virus or also called the hepatitis delta virusoid because it's like a virusoid. So it's much larger than the plant virusoid, but HDV has a circular single-stranded RNA. So again, our single-stranded RNA, non-self-replicating, its genome is larger at 1,700 nucleotides versus over here the 220 or so. And it can direct the biosynthesis of HDV-associated proteins. So this is one that actually does make proteins. And this is in humans again. So the HDV helper virus is the hepatitis B virus, so HBV. So when a person has co-infection with the hepatitis B virus and our hepatitis delta virus, or hepatitis delta virusoid, it results in a more severe pathological changes in the liver. So it can be a more severe infection, which is how this was found. That's what it says down here. This is how it was discovered. So some people had much more severe changes in their liver, and then they took a look at it, and then they noticed that there was this virusoid that was kind of taking a little ride with the hepatitis B virus. So there is a hepatitis B virus that is separate from the virusoid. People don't have as severe pathological changes with that hepatitis B virus on its own. It's just when it's packaged with the virusoid, then it can be much more dangerous.